Good morning and welcome, especially to the computational biology and biostatistics students. My name is Jeff Gruen, and in our lab, we study the genetics of learning disabilities. So I'd like to present to you this morning rotation projects available for you as graduate students in the fall. Our lab works on learning disabilities, and we focus on dyslexia, and for good reason. Dyslexia is, by the way, a reading disability. And the reason we focus on it is that worldwide, about 10% of students struggle with dyslexia. And these are students in regular classes, at regular schools, and with normal or above normal IQs. Between 60 to 80% of reading performance is due to multiple genetic variants. In other words, this is a highly heritable trait. But for some children, a durable intervention can make a huge difference in both their academic performance and in their lives. So this is data I'd like to present to you from my colleague Maureen Lovett from the University of Toronto, where Maureen looks at randomized controlled trials of intervention for children she identifies as struggling with reading in school at different ages. So here's first grade, second grade, third grade. And when she identifies these children that are struggling with reading, she offers a highly comprehensive, intense reading intervention program, which is administered generally over the school year. And then she tests their performance once again 2 years after completion of the intervention. And what you can see by her graph and by her data is that in grades 1, 2, and 3, 2 years after this intense intervention, about 75% of those children will be reading at grade level. But if the children are identified later in life, in middle school or high school, you can see that only about a third of these children actually responds. So we can learn from Dr. Lovett's data that certainly early identification is really critical, and as well as early intervention. So since reading is a highly heritable trait, our lab has been involved in generating and creating a genetic screening panel to identify these kids at an early age, even before psychometric testing is very accurate. And so we do this, of course, by getting saliva from these children and then extracting DNA from that and looking for DNA markers. So to identify children at risk for dyslexia at the age when intervention works best, at the early ages, so we have been developing a genetic screening panel. We do this through genetic association, genetic linkage analysis, deep sequencing, and through rare variant analysis. And over the years, we've published this in various journals, either on data that we've generated ourselves or in collaboration with others. But at the heart of all our studies are the children themselves. This is a photograph actually extracted from the New Haven Lexicon Project. These are children in one of our intervention programs. And these really formulate the basis of our work. It's always translational work where we start in the classroom with children, and then we use the data that we can extract to be able to identify those genetic variants that seem to be involved in the process. And so you can see here by our approach in the lab is that we start with clinical samples, and then in this case, we do whole genome sequencing here at 30X in New Haven Lexicon Project. And then we analyze the data using a variety of approaches: whole genome sequencing, GWAS, linkage analysis, copy number variant, polygenic score, and several other different ways. Then in our sort of multidisciplinary approach, we can apply those variants, the findings that we see that we acquire by MRI, both in our data sets and in extant data sets. And here we look at both structural and functional, and I'll review some of that data with you. And then those genetic variants that we identify, we then recapitulate or model in human embryonic stem cells that are then differentiated into neurons or brain organoids. And then ultimately, the idea is to bring it back into the classroom itself. So when I think about computational rotation projects for CB&B graduate students, they're really in 7 areas: in genome-wide association studies, DNA sequence analysis, genomic analysis, genetic analysis, imaging genetic analysis, RNA sequencing, and evolutionary genetics. And I'd like to start off by really looking first at these genome-wide association studies and how we apply the data to that. So GWAS, of course, is the cornerstone for identifying common variant association for polygenetic traits and disorders, including learning disabilities. There have been over 5,000 GWASs that have been published. They generally account for a small fraction of SNP-based variants, and for any single SNP, it's a high bar to achieve genome-wide significance after adjusting for multiple effects, also known as the Bonferroni significance bar. So in general, GWAS requires large data sets. So let's look at some of the data sets that we've examined and others have examined, and it's available for study. And what I'll do is I'll look at 10 data sets, and we'll examine them for the number of subjects, as well as the specificity for reading and language variables. So we can start off with our own homegrown project called the New Haven Lexinome Project. It's a longitudinal 7-year project. We look at 2 serial first grades out of several New Haven public school elementary schools. We do serial assessments. We look at deep reading variables, deep language variables. As I mentioned, we do 30x whole genome sequencing, but the numbers are small. It's about 440 subjects total in our latest analysis. So here, what you can see is on the reading language specificity, it's really, really strong, but the New Haven Lexinome Project is relatively small in the number of subjects. Let's contrast that with 23andMe, a recent publication we currently have under review. Here we have over 1 million subjects, but there's only a single question about reading or reading disability. There are no assessments, and it's a microarray study. And so it gets very high under number of subjects, but as you can see, it's very low in reading language specificity. Probably similarly with the UK Biobank, here we have over 200,000 subjects, generally greater than 40 years of age. And they're probably amongst the very few variables available to study is educational attainment, which is sort of blunt and doesn't really correlate well with reading performance. There is a yes/no question on attention, and there are no assessments. So again, it's high on the number of subjects, but it's low on reading language specificity. The Psychiatric Genetics Consortium, which is about 30,000 subjects, again, they're mostly adults. Again, there's no assessment. So it has some power or has some number of subjects, but again, low on the reading language specificity. This is the Gen Lang Consortium. It's a very interesting consortium out of Europe, Australia, and the Americas. And so it has a combined data set of about 30,000 subjects, and they range in age from 5 years to 20 years with multiple reading and language assessments. However, they're in multiple languages and they don't completely overlap. And so it's sometimes difficult, to combine these data sets so that the number of subjects is pretty good. Specificity is a little off because not every variable is assessed on every different subject, partly because of the different PIs that are combined here, and also partly because of the languages. Next is the Avon longitudinal sample of parents and children, the longest longitudinal study in the world on children. There are now 15,000 children in this data set, and they've been followed from 0 to 32 years of age. And there are multiple reading and language assessments. And so pretty high on the specificity scale, but the number is relatively small. Next, we have the ABCD study, the Adolescent Brain Cognition Development Study, which is out of the NIH. It's currently ongoing. There are about 10,000 children in this study. It's longitudinal between 9 and 15 years, but the assessments are through the NIH toolbox, which isn't very good really for assessing reading and language. So again, it's on the subject number, it's small, and on the specificity scale, it's probably midway. Next is the Philadelphia Neurocognition Consortium. This our cohort. This is about 9,000 children ages 9 to 15 years, and there are no reading or language specific questions, although there is a vocabulary test on this. And here, so the specificity is low, and the numbers are low. And then we have the GRAD study. It's a homegrown study, the Genes, Reading, and Dyslexia study out of Yale. Here we had 1,350 subjects ranging in age from 7 to 16 years, multiple reading and language assessments. It's a mixed cohort of exclusively Hispanic Americans, and African Americans remains the only genetic study of dyslexia in these underrepresented minorities. So here, high in specificity, but low in the number of subjects. And then finally, the Pediatric Imaging and Neurogenetics Consortium of 1,250 children ages 3 to 21 years. Again, they used the NIH toolbox, which It doesn't really probe in-depth reading and language assessments into mix of multiple ancestries. And so the ping sort of gets midway and has a relatively low number of subjects. So as you can see here, this is a balancing act between power and specificity. And so here, the cohorts that have the largest, the most amount of power often have the least amount of specificity, and likewise, the cohorts that have high specificity actually have generally low power. So we can leverage, though, the power of these cohorts in several different ways by combining them or working with them or indexing them with larger data sets. And we can do this in a number of different ways. One was we can constrain the search space or the number of markers. We can look at gene-wise effects, candidate genes. We can look at locus-wise effects. And we can do methods that look at marker ranking and selection. We can combine data sets here on the low end to make them stronger. We can also use the polygenic risk score to condition a score from the larger data sets and use that to target the smaller data sets. And then, of course, we can look at genetic sharing between the two. We can also play to the strength of the specificity. And so therefore, we could look at variables with large genetic effect. And to do that, one way to do that is to look at imaging genetics. And so we can use the fMRI or the functional magnetic resonance imaging data in order to, which has a very large effect, and then use that and condition our genetic studies on that. And so what we do is we predicate our genetic analysis on human brain MRI. We could do structural, we could do functional connectivity, and we've published on this as well. Here is a genetic marker called READ1, which we showed influences functional connectivity between right and hemisphere brain structures for critical reading and language. And so here you can see a well-known language network connected by 4 spokes and consisting of 5 nodes. And this particular connection here between the right inferior frontal gyrus and the right superior marginal gyrus, that connection highly correlates with the READ1 genotype. And this is a well-known, and it's critical for fluent reading as well as strong language ability. And we published that back in 2018. So this is an example of some of the imaging genetic analysis where we predicate our genetic analysis on really a very strong variable. More recently, we've been working in Amazon Web Services in the cloud in order to do our imaging genetic studies up in the cloud. Until then, we've been doing it on the high performance cluster. And here you can see a new program that we're currently developing called NeuroStack, which is an AWS cloud formation tool that sets up the infrastructure required to perform neuroimaging and genetic analysis using AWS cloud computing. And within this structure, we can do lots of different programs. If you're familiar with imaging, we can do things like AFNI and FreeSurfer, but from the genetic side, we can also do our Plink analysis and use other genetic tools right within the cloud and not have to download to their high performance computing cluster to do the analysis. At the heart of this tool is the Lambda scheduler. What Lambda can do is it can really minimize the cost as best as possible for doing these analyses within the EC2 instance. And so it's really a plug and play sort of program, so you don't have to do any, it's more of an app, it's less of a program, so you don't have to do any programmer wrappers whatsoever, and sort of plug and play for the uninitiated. So we can go from genetic information index into functional brain, or how things actually work, and then we can recapitulate those variants that we find in human embryonic stem cells that are differentiated into neurons and or brain organoids. And so we've been doing that over the last couple of years, where we look at knockdown of a well-known dyslexia gene called KIAA0319 and its effect on the on transition from human embryonic stem cells to neuroepithelial cells, which in control cells are hallmarked by strong expression of SOX10. And then when we knock down KIAA0319, it actually decreases SOX10 expression. And the transition to neuroprogenitor cells, which is usually highlighted by expression of PAX6, actually comes early. And now we're in the process of doing RNA-seq and RNA-seq analysis for each of these steps and each of these intermediate steps in the transition from human embryonic stem cell all the way to mature neurons. So that's another computational biology program project that's currently available within the lab. So beyond that, of course, is how do we bring it back to the clinical situation there? We've been looking at evolution, not of humankind, but of the evolution of language. And here's data from a paper that we published back in 2000 on a particular genetic variant called RU1-1, where we looked at different populations with very different languages. Here it is an Igbo population from Africa, and you can see by the size of the sphere, they have a high frequency of this RU1-1 genotype. And they also have a large number of consonants in the Igbo language. In contrast, when we look at a population of Japanese speak in that language, you can see that the frequency of RU1 is much smaller because it's a smaller sphere. And because of the color, you can see that the number of consonants is low. And we did this with 60 different populations from around the world, speak different languages, both transparent, non-transparent, Western, and Eastern languages. And what you can see here is that if we look at the comparison of vowels and the frequency of RU1-1, they don't correlate very well, but they correlate very nicely with consonants. So it appears that the acquisition of consonants into a language may be partly in effect, depending upon the RU1-1 allele that population has in high frequency. So as you can see, we've now come for a full circle back into the clinical part of the lab to enable our early screening to identify children with reading disability, and that's an ongoing project. So just to summarize, there are 7 areas where rotation projects are available. They're available in GWAS, DNA sequence analysis, genomic analysis, genetic analysis, imaging genetics, RNA sequencing, and evolutionary genetics. If anybody has any questions, this is my email address. I'd be happy to answer any questions you may have, and welcome meeting you.